Welcome to the Med Evidence Podcast, hosted by Dr. Michael Koren and Michelle McCormick. Med Evidence, where we help you navigate the real truth behind medical research with both a clinical and research perspective. In this podcast, we'll have discussions with physicians that have extensive experience in patient care and research. How do you know that something works? In medicine, we conduct clinical trials to see if things work. Now, let's get the truth behind the data.
This is Med Evidence, Truth Behind the Data, with Dr. Michael Koren. He is a practicing cardiologist and Chief Executive Officer at ENCORE Research Group, which conducts trials across Florida, clinical trials across Florida. He has been the principal investigator of over 2,000 trials and has been published in the most prestigious medical journals. Dr. Koren received his medical degree, cum laude, at Harvard Medical School. For more information on local trials, visit encoredocs.com. That's ENCOREDOCS.com or call 904-730-0166. Dr. Koren, we learned a lot about the beginnings of clinical trials. We are still waiting to see how the lady with the tea experiment worked out, so I'm patiently waiting for that.
Sure.
But in the meantime, we've talked about truth. Mm-hmm. We've talked about the first trials with a vaccine for smallpox, 6 people, kind of a small trial, but you know, what's next? How did the trials grow since 1721?
Sure. So again, looking for truth, which is what this is all about, the folks that did the experiment in 1721 at the Newgate Prison became convinced, based on this relatively small sample size, that they had a solution. And again, it was prospective. It wasn't just somebody's story. It wasn't an old wives' tale. It was actually...
It seemed to work.
An experiment. There's a couple of interesting things about that. There were actually... I talked about deductive reasoning, and the key elements of deductive reasoning is that all the inputs to the deduction are true, and the question is framed properly. But how do you know that all the inputs to the deduction are true? So there's actually some very interesting little sidebar of the Newgate Prison story, which is that one of the prisoners lied about their history.
Oh. One
of the prisoners actually had smallpox before and didn't disclose that.
Huh.
And they only found out about that because when they tried to inoculate that person, the pus would not make a lesion on the leg because that patient's immune system was already working and fighting smallpox. So that patient had to go back for a second surgery with a bigger cut and more pus before there was any evidence that there was some problem.
Okay.
So with any experiment, there's always people that may cheat, and you have to be careful about that. But if there's very rigorous design and a lot of care taken in terms of how you set up all the details, then you will come up with a deductive reasoning conclusion. The other thing that's interesting about the Newgate Prison story is that they actually tested the patients after the fact by having some of the people that were in the experiment sleep in the same room with somebody with smallpox to see if they got sick. Mm-hmm. And they didn't.
Yeah, that was going to be one of my questions is even if they were injected or the pus was put in, are they still contagious to the other population?
Well, once they get past that initial response, they are immune, and then they won't get the virus or spread the virus.
Okay.
Which is true to today. And that was our early learning about that. But anyhow, that was a small study, and we needed more confirmatory evidence, of course. So the next big piece of the puzzle actually occurred in the same year for the same pandemic. Obviously, when you have a virus, it spreads. We know that. And even though travel was not as extensive in 1721 as it is today, people still traveled. So there were ships going back and forth between England and the colonies all the time. And what if they brought diseases? So in 1721, during this pandemic year in Britain, there was a ship that came over, and all the sailors were actually healthy when the ship left. But as the ship was crossing the ocean, one of the sailors got sick with smallpox. And people started freaking out, of course. So they get into Boston, and they know that there's a sailor there that was sick. And the word spreads in the community that there's a sick smallpox person. So there's a fellow named Cotton Mather who was a minister at that time, and he owned a slave named Onesimus. And Onesimus was a really smart guy, and Cotton Mather and Onesimus kind of had a little bit of a friendship going. And Onesimus told Cotton Mather that in Africa, where he was born, what they did to prevent this was to do inoculations. And Cotton Mather was open-minded to a degree and said, well, that's interesting. Maybe we should be doing this to protect people in Boston, which is where he lived and where that ship came in. So anyhow, Dr. Cotton Mather, who wasn't, quote, a really physician, he was one of these Renaissance people that did a little bit of everything, but he liked to think of himself as a man of science. He found this guy named Dr. Boylston. For those of you that have been to Boston, there's a Boylston Street. I was just there on a very nice street, by the way.
Okay, all right.
Lovely street, and it was named after this doctor.
Okay.
And they together, based on Onesimus's advice and his guidance, inoculated 287 people in Boston in 1721 by making cuts in their leg and putting smallpox pus in it. And lo and behold, the people that were in that group of 287 had a risk of dying that was less than 2% versus about 16% for the general population. So incredible difference was made even in this very primitive type of inoculation.
Okay.
And so that led to this concept of inoculations and vaccinations. And ultimately, the observation was made that not only do previous smallpox victims seem to be protected against smallpox during pandemic years, but also people that get cowpox. So they found that milkmaids who were constantly being infected with cowpox, which is a mild infection, seem to be immune from smallpox. And then Dr. Jenner in the UK eventually created a smallpox vaccine based on giving people cowpox, which is less serious type infection than actually inoculating people with smallpox itself.
Yeah.
And then of course things developed more, and we developed a whole industry based on large-scale clinical trials once we understood statistics better. And that's getting back to R.A. Fisher, who created the infrastructure, the intellectual infrastructure for statistical analysis in the early parts of the 20th century. And then studies like the Framingham study and other studies that became interventional were able to show the benefits of cardiovascular disease prevention, cardiovascular risk factor disease prevention, and also the benefits of inoculation. So a polio vaccine being a great example of that, where we literally wiped out a scourge, horrible thing. Obviously, smallpox has been completely eradicated based on a vaccination strategy. And most recently, we had this massive effort for COVID-19 showing that literally that we can, one, develop a vaccine in an incredibly short period of time with modern technology, two, inoculate tens of thousands of people, and three, do the hypothesis testing required in an incredibly vigorous way where we know exactly how effective this thing is. We know exactly what side effects to be worried about. And we know that there's going to be mortality differences and differences in hospitalization based on these interventions.
Yeah. Do you think that COVID-19 vaccine, are the trials still ongoing?
Sure. Yep. Well, thank you for asking the question. And obviously the wonderful people who volunteer for the trials in the first place are still being followed. Good. So we appreciate them. Obviously, in the beginning, they were involved in a prospective, blinded, randomized trial with a placebo arm. So the early people that participated in it didn't know for sure if they were going to get a real vaccine or a placebo. Eventually, everybody was crossed over. So again, they stayed blinded, but the people who didn't get the vaccine initially got it after a few months. Yeah. And that's what's called a crossover design. And that's a concept of fairness, that everybody eventually gets it, but there's a time lag. So you can see if the people who got it earlier do better than the people who got it later. Did
the boosters kind of like piggyback on that vaccine, initial trial?
Right, and then there was the booster trials, and we participated on the booster trials here in Northeast Florida and other places, and that showed that the third dose was also very effective. People who got their initial vaccinations, the two-part series for either Moderna, Pfizer, or Novavax, they were protected, but after a period of time, that protection wanes. So they're more likely to get infected with COVID after about 6 months. Now, it's a much less severe illness, but nonetheless, they still have some vulnerability. So giving a booster, again, reduces that vulnerability after a 6-month time lag. And what was very, very interesting, and this gets into this concept of faith and truth, is that we were just proving that the third dose worked, and the FDA approved a fourth dose based on faith. So they got to a point where they said, okay, well, We know the vaccines are safe and effective in the beginning. We know that a booster works, and a booster seems to kick in and it becomes necessary for high-risk people after 6 months. So there's no reason to believe that a fourth dose after another 6 months should also be beneficial. But the crazy part about this is that the FDA approved, quote unquote, the fourth dose as the data were coming out for the third dose. So even though it was kind of known that the third dose worked, the formal presentation of the data, which was a New England Journal paper that I was part of, didn't even come out until the FDA was having discussions about the fourth dose. And some people in the scientific community were actually upset with the FDA by jumping the gun.
Right.
They're saying is, okay, we're all about deductive reasoning, we're all about clinical trials, and you're making an assumption rather than looking at things from the standpoint of certainty.
How interesting. Do you think this will become a yearly flu shot, type of situation for folks to just continue
I personally
to...
think so. I do think that we know that viruses mutate. We know that certainly coronaviruses mutate pretty rapidly, and COVID-19 has mutated. It seems that it's fortunately mutating to a more benign form. Thank God for that. But who knows? It can mutate to something else that may not be quite as benign. So we have to be on the lookout for it. And we know that our immune systems are incredibly capable of fighting the virus in the 6 months after you're exposed to the vaccine. So it only makes sense that we'll use a strategy like we use for the flu and other things, where we can hopefully give the vaccines on a regular basis to people who are at high risk and prevent them from having severe complications. Now, there'll be some discussions about who's high risk. There'll be probably high risk definition based on both your exposure risk, so people in the healthcare world, for example, or people that have are first responders, et cetera, and people who are very, very vulnerable if they were to get infected, cancer patients or people with diabetes, or et cetera, et cetera. So we'll look at that over time. But that's also true for other viruses. So COVID-19 was the talk of the last few years, but we have some other nasty viruses out there. Chikungunya, we're doing studies on that. That's a problem in tropical areas that's now coming to Florida, and we're doing work on that. RSV, respiratory syncytial virus, is a virus that can be as nasty or worse than COVID in some people, and will kill vulnerable people. Yeah. Flu, of course. Some years the flu seems to be more pathogenic than others for reasons that are not always completely understood, but the flu is a virus that mutates. It's a virus that, if you're vulnerable, can put you in a really bad spot. And so as we speak, we're doing studies about all these things. We know vaccinations work, and we're trying to get to some of the nuances of the strategy. So once you know that the vaccines work, then the question is, okay, do you have to change the vaccine each year? Or is a basic vaccine important? Should it be every 6 months? Should it be every year? Should it be every 2 years? Certain vaccines we give less frequently, or some we give more frequently. So vaccine against pneumococcal pneumonia is typically given every 5 years, and that seems to be enough to keep the immune system on guard. Shingles vaccine is another example of that. Shingles is based on a virus that we get typically as children.
Chickenpox, right?
Yeah.
But now there's a chickenpox vaccine too.
Yeah,
so I wonder if we could just stop there real quick with the chickenpox, because my kids got the chickenpox vaccine. I never had it, but I don't have a shingles vaccine, but I had chickenpox.
Right.
So I'm probably more vulnerable to shingles versus my children who have had the vaccine.
Right. No, absolutely.
As they age, yeah.
Right. That would be expected. Actually, one of the rationales for giving the chickenpox vaccine rather than getting infected is that the virus can live in a dormant stage in your nerves. So if you've actually been infected, it's probably still in your body, whereas your kids who are vaccinated probably won't actually have that in their nerves. So that hopefully that'll prevent them from getting shingles down the road.
Right. So your shingles vaccine, is that ongoing?
This is the learning of clinical trials.
Yeah, it's fascinating.
This is how we establish the truth. And again, through this process of deductive reasoning, using deterministic statistics and prospective, blinded, and randomized studies, we're able to figure out really what's going on in the world.